Absolutely.
Thank you, Dr. Jordan, for the introduction and for really helping to organize this meeting today. So my task is to talk about one of the most exciting areas of medicine now, which is what I call gene-targeted therapy and how it applies to neurofibromatosis and schwannomatosis. I hope I'm successful, but if this lecture is not, please let me know. I'm trying to figure out a good way to show this content to people in our clinics, and let me know if you think if it worked. So there's a couple of important disclosures here. The most important is that I'm a co-founder of two NF-related companies because we're trying to bring new medicines to persons with NF1 and NF2-related schwannomatosis. And one of them, NF2 Therapeutics, is actually a gene therapy company. So you should know that as my conflict as part of the disclosure today. So here's what I want to talk about. We all come to this topic with different levels of education. And so I want to go over a little bit of what I call genetics 101. And I'll do my best to bring us to a basic place to understand gene-targeted therapy. I'm really not going to spend much time talking about NF1 and schwannomatosis clinically because we'll be getting a lot of that throughout the day, and because for reasons of time, we won't be able to go in depth. But then I want to bring you up to speed on where we are in gene-targeted therapies in 2023. And really close with some of the opportunities and challenges that presents to us as we think about NF1 and schwannomatosis. So I don't know if this is big enough, but if we start from the beginning just to understand genetic conditions, the first thing to understand is what a gene is. And this goes back to many of us in high school, where it's hard to actually put it into words, but the way the New England Journal of Medicine does, it says a gene is the fundamental physical and functional unit of heredity, meaning this is what makes us look like our parents and our children. It's what we share genetically. And that gene is, as you can see in the figure on the left, all this genetic material resides in chromosomes. We have 23 pair of chromosomes and sex chromosomes in our cells. And those chromosomes are composed of DNA. And that DNA, let's see, can you see it? No. See, how do I get a pointer? One second. That DNA, these are the chromosomes. This is what the gene structure looks like. But on a fundamental level, this is composed of different molecules called nucleotides that are ordered in a very specific way. And those genes can encode proteins that are really what makes our cells work. Now, to understand genetic conditions, you have to understand what a genetic variant is, meaning something that varies from a standard. So there is something called a reference genome. And what the reference genome is, they take DNA samples from many, many, many people. So this is not from a single individual. And they look at the sequence here and they come up with a reference, which is the standard for human beings, right? And it's something that's been developed over the last 30 years. And the reference genome is what we will point to for every person who gets genetic testing done, right? People like us. And so when you think about genetic variants, we use an analogy or a metaphor of spelling of words. So if you look on the left, and and you have a word like neurofibromatosis written out, if that's your reference, right, what your normal is, think of the changes you can make. So if you take the B in neurofibromatosis and make it into a Q, you get a spelling you get a misspelled word. You can also have genetic changes that stop the genetic sequence. And so we call that, you can call that a type of variant called a nonsense variant, where it just stops reading. You can have deletions where you lose a couple letters. Here you lose ROM and you can see the misspelled word. You can have inversions. You can have things flip in sequence and you can get a misspelled word that way. You can have insertions or additions to the letters, and you can see that you get a misspelled word here. And the same way you can get duplications. And I didn't want to spend a lot of time. This is where we often get bogged down as doctors trying to explain this, is we go into all these details about what can go wrong with the genetic code. It's not important for this talk as much as to point out that there are many, many ways where our genetic code can get mixed up and create variants that can cause different diseases. And so if you take a step back, what is gene-targeted therapy? So from a doctor's point of view, it's a medicinal product. So it's something that goes through the drug process that's used in or administered to humans to regulate, repair, replace, add, or delete a genetic sequence with the aim to treat and cure disease. And the focus here is on genetic sequences as opposed to on the proteins, because most of the medicines we use currently are focused on proteins and not on these two things here. So this figure is to remind us how information flows in a cell. It flows from the DNA, which is what makes up the genes, to an intermediate called RNA, which then gets translated into proteins. And without getting bogged down in the science, I would say gene therapy is focused more on the DNA and RNA side, whereas many of our current medicines are focused on proteins. And so to reiterate something that we know in this room, both NF1 and schwannomatosis are genetic diseases. That is, they're caused by genetic variants. And we know that the core issue with NF1 is the presence of nerve sheath tumors called neurofibromas. And the core feature of schwannomatosis, including what we used to call NF2, now called NF2-related schwannomatosis, and other forms of schwannomatosis, is the presence of schwannomas. But as we all know in this room, in addition to those two tumor types, there are multiple other things that can happen, whether you have NF1 or whether you have schwannomatosis. And this is just a summary. And I'm not going to go through this because I'm going to let other speakers talk about many of these other findings that can happen in these two conditions. Now, why are NF1 and schwannomatosis so amenable to gene therapy? Why are we so excited about it? Well, I would argue the reason that these conditions are attractive targets for gene therapy is that they are monogenic disorders. What does that mean? That means both of them are caused by a genetic variant in a single gene. That's different than diabetes. You may live in a family where diabetes runs in your family or high blood pressure runs in your family. Those conditions have a high hereditary factor. That is, it runs in families, but neither high blood pressure nor diabetes are caused by single genes. They're caused by multiple genes that we have. NF1 and schwannomatosis is different. They're caused by single genes. Now, they're not exactly the same. You can see in this representation, NF1 is caused by genetic variants, misspelled genetic variants in a gene on chromosome 17 called the NF1 gene, as you know. Schwannomatosis is a little more confusing. We won't go into all the details. We've identified at least 4 genes, all on chromosome 22, that cause schwannomatosis. But to be clear, for any individual and any family, it's caused by a single gene there. So you're either a family that has these genetic variants in the NF2 gene, or in the SMARCB1 gene, or in the LZTR1 gene. And so these conditions, I think, are attractive because they're caused by a single gene. Now, what are these genes? These genes are what we call tumor suppressor genes. What does that mean? Now I don't want you to read the details, but if you go back to your early biology, you'll know that cells, each cell lives in a cell cycle. Sometimes the cell's dividing, sometimes they're not. We know that the process of cell division, as occurs here, is highly regulated. And there's an interplay between genes that promote cell division. We call those oncogenes. That's equivalent to the gas pedal. And at the same time, we have the balance of tumor suppressor genes. These are genes that prevent cells from dividing. And so as we think about what can go wrong in these genes, when you lose the normal function of a tumor suppressor gene like NF1, like NF2, like SMARCB1 and so forth, you're basically losing control of that cell division. And what happens is that if a single cell loses both copies, that cell can form a tumor. And that's what's common across all these conditions. So why do people get so many tumors? This explains why. We all have two copies of every gene, one from our mother and one from our father. And if you have a pool of susceptible cells, random genetic events can inactivate one copy of a gene like NF1 or NF2. It takes a second random genetic event in the exact same cell to lose a second copy. And what you get in that case is a tumor. And if you're not born with NF, if you're somebody who doesn't have any of those issues, that's how you get a random tumor. So people can get neurofibromas and not have NF1. You can get a schwannoma and not have schwannomatosis. And this is how that happens. But more commonly, for conditions like NF1 and schwannomatosis, people are born with one functional copy of the gene and one non-functional copy. Therefore, your pool of susceptible cells that can form tumors is much larger. And what you can see here is that the random genetic events, somebody's advancing this, random genetic events then fall on cells that already have one abnormal copy. And what happens is you get multiple tumors. And what I'm trying to display here, we now know, and this is an example from vestibular schwannomas in NF2, that what we see as a single tumor affecting the hearing and balance nerve is in fact the accumulation of tons of little mini tumors that all form independently. And we believe now that this process, this susceptibility to multiple tumors is what causes all these tumors across NF1 and schwannomatosis. So I am not a structural engineer and I know nothing about bridges. But what I want to show you here, this is directly from the Encyclopedia Britannica. What I want to show you, and the metaphor I want to make to you is, as we think about gene-targeted therapy, there is no such thing as one type of gene-targeted therapy. And I think that's a common misperception. And so I want to explain that with using the metaphor of bridges. There's multiple types of bridges, and you can see the ones here. It's a beam bridge. It's an arch bridge. Why do we have so many types of bridges? We have so many types of bridges because the bridge type is selected for the span that you're trying to cross. So a certain type is good for each of the different conditions here. And as I told you, I don't know anything about building bridges, but I think you understand what I'm getting at. And so I would propose we think about gene-targeted therapy the same way. And there are three major classes, and this is a simplification. On one hand, you can think one type of gene therapy is when you want to add a gene. For example, if the problem is you have a non-functioning gene like NF1 or NF2, then adding a functioning gene can repair that problem. A second type of gene-targeted therapy would be gene silencing. And in that case, you have a gene that is producing a toxic protein. And there are many conditions like that. The solution then would be to use gene therapy to reduce that protein. A third type of gene therapy, targeted therapy we talk about is called gene editing. This is all the rage now. You may have heard of something called CRISPR or CRISPR-Cas, and this holds promise for curing so many conditions. And the way that gene editing works is by correcting the misspelled word. Imagine a pair of microscopic scissors that could cut out the faulty gene and put in the correct one. Now, I would argue, just like with bridges, the choice of gene therapy depends on what gene you're working on, what type of genetic variations you have, how much it costs, and so forth. So here's the challenge. This is a schematic of the NF1 gene. and the identification of all the genetic variants in 339 Korean patients. And what you can see here is this is the gene, a schematic of the NF1 gene. These are all the variants. What this shows you is that there are no hotspots here. It's not like there's one location with one type of genetic variant that has to be treated. And so for everybody here who may have gotten their own genetic testing, you come back with what we call a private alteration. That means it's for you and your family and probably not for anybody else. There are some very rare things that are shared, but in general, they're not. And what I wanted to highlight here is you can see there's a broad range of misspelled words, what the mechanism is. So the challenge here is that there's no hotspots, that there's a range of different alterations, and that it goes across all of the parts of the NF1 gene. And that really is the challenge as we think about gene therapy. Don't know why it's advancing for me. We might have to work on that from the side of the AV people. So let's transition now into gene therapy. On one hand, it seems like it's been here forever, but practically speaking, it's new. It wasn't until the 1970s that people even began to think that we could replace or supplement the lack of these DNA areas. So that was just formulated in the 70s. We learned in the 1980s that we could use viruses to actually transfer a gene into a cell. In the 1990s, the first human trials were initiated, but these were limited by some very significant problems, including deaths of individuals participating in human trials. And rightfully so, the FDA, the NIH, and other groups halted clinical trials for many years. But by 2004, we had our first approval. Now, you're not going to be able to read this, but I want to show you this is the age of gene therapy. There's no question. Beginning in 2004, in China, was the first approval for a gene therapy to replace the p53 gene. And I won't go through these in detail, except to show you in this column, I've listed A's and O's. A is gene additions. 86% on this page of these replacements, of these gene targeted therapies were gene replacement, which is very important because it's the same problem that we're struggling with for NF1 and schwannomatosis. Continuing now, this is the other half of the gene therapies that are currently approved. And again, the vast majority represent gene additions. I'm not going to talk about oncolytic viruses. It's another interesting area for us to think about, but not something we're working on right now. So I've talked about those gene targeted therapies were focused on DNA. There's a whole nother group of gene targeted therapies that are focused on RNA. And here are some examples of those and the conditions. I guess maybe I'll go back and just say for the previous conditions, many of these are tumors, focused on treating tumors, but some of them are focused on other things like blindness or a topic I could have spoken about all day, spinal muscular atrophy. And in RNA therapies, we can see a lot of these have to do with either amyloidosis, which is a genetic condition, or muscular dystrophy. And what I wanted to point out is that there's 4 therapies that focus on exon skipping, and I'll come to that in 1 second. So there's a lot of approved therapies, not enough, but a lot. And so the trends as I see it is that 24 therapies since 2004, the majority are focused on gene addition, which is directly related to what we want to do in NF1 and schwannomatosis. And I would point out that we have therapies that replace tumor suppressor syndrome. So there is an obvious path for us to move forward. Of the 15 RNA therapies that are approved since 2013, 4 of those specifically target an increase in protein levels, which is what we want to do for NF1 and schwannomatosis. So we have the availability both of DNA therapies and RNA therapies, at least conceptually moving forward. So now I want to transition into what's going on in NF1 and schwannomatosis. And I want to assure you that multiple labs are working on this problem right now. And here are just 3 examples so that you know that on the left, the Children's Tumor Foundation has a large effort in gene therapy. So does the Gilbert Family Foundation in the middle, as well as NF2 Biosolutions. And I'm not going to go through the details. It's not worthwhile at this point. But these laboratories are funding, these agencies and groups are funding many labs to work on this problem. So what are the key problems in the last few minutes? For the doctors and scientists working on this, we know that no single approach is going to work for every patient because of that range of genetic variants we see. We struggle with how large these genes are. How are we going to replace genes like NF1, which is enormous? How much of the NF1 protein do we have to replace to make people better? Is it 1%? Is it 10%? Is it 100%? What happens if you get too much protein? What if you have 150% or 200%? Will that hurt somebody? If you replace these genes, how long does that last? And what is the effect both on tumors, which is the problem we think about usually at the beginning, but also the normal tissues, or what is the effect on things like learning disability? And then clinically, how can we administer these therapies? Is there a window for treatment? Should we be treating children or adults? Which of those many manifestations should we treat? And which patients should we enroll in trials? And finally, what endpoints should we be used? Should we use to see if something is active? So I just want to bring up quickly some of the key safety issues. We know from those early trials, there's a risk of cancer in some individuals when you're inserting DNA into genes. We also don't know the effect if we overproduce these genes. I mentioned that. And then finally, what are the immune responses, which can either minimize the benefit of these treatments or even prevent people from getting treated a second time? We know that long-term surveillance will be important. And so I quickly want to say that on the clinical side, the REINS group, which stands for the Response Evaluation in Neurofibromatosis and Schwannomatosis, has a dedicated working group that is working across the world to understand these issues about how to move forward. And so the big question we're struggling with in REINS is who should participate in these early trials when they happen? Should it be somebody who's severely affected, who has limited options? And so they say, yes, I would be willing to try something that may have risk. The question is, can gene-targeted therapy work on persons who have advanced or severe disease? And how will that prevent us or not from learning about the risks? Maybe the problems you see in those trials will be due to people who are sick rather than the actual gene therapy. Well, how about treating mildly affected persons? Well, the problem is they have more treatment options and therefore may be less willing to accept risk. And mildly affected persons are more likely to tolerate treatment. And to summarize that scenario, these individuals have more to lose and more to gain. So we're struggling and we're working with patients and other family members to help figure out what's the right way to move forward. So this is my last slide. So what are the prospects of gene-targeted therapy in NF1 and schwannomatosis? This is my own personal belief. I think there are many hurdles. We have to acknowledge reality. But I am extremely hopeful that based on where we've been since 2004 and the science and technology that's available, that this is around the corner. And I think the goal, of course, is for us to be involved in this process. And I know everybody, and our programs at Children's and Mass General are deeply involved in this work. And so I would call this realistic optimism. But I don't want you to think that this is here next year. And this is really important to me. We're trying very hard, but there are many steps before we get there. And people always ask me, Scott, when do you think this is going to happen? I don't think it's the next 5 years. I'll be very honest. I think it's further than 5 years because there's lots of work to be done. So I'm very optimistic. And at the same time, I don't think this is around in the next few years. So with that, You can find me afterwards. I've used up my time with 13 seconds remaining. And if you have questions, I'd be happy to answer them afterwards. Thank you.
Thank you very much, Dr. Plotkin. Okay.
Yeah.
That was really fantastic.